Well, thank you, Chris, for the introduction, and welcome everyone to this session where we'll be discussing exciting work in the therapeutic development for inflammation and immunology. With our four panelists, Natalie Hollis, Kevin Giudice, Rosanna Capeller, and Andre Turenne, we will focus on approaches for small molecule development in a therapeutic area that has been dominated for the last quarter century by biologics. Although our session is titled Oral Formulations, we are really focused on the contrast between small molecule drugs and biologic therapies. Small molecules are synthesized compounds, typically less than 1 kilodalton in size, and typically administered orally. Biologic therapies are secreted by living cells, most commonly monoclonal antibodies or fusion proteins, and are typically administered parenterally, intravenously, subcutaneously, or intramuscularly. Biologic therapies have little intracellular penetration and are most often used for targeting soluble molecules in the circulation or molecules that are bound to the cell surface membrane, while small molecules typically target intracellular targets. Historically, our first modern therapies have been small molecules for autoimmunity and inflammation. Corticosteroids were introduced approximately 1948, that's 75 years ago. Methotrexate, azathioprine in the 1960s, cyclosporin, and a host of others have dominated the field until approximately the late 1990s with the introduction of rituximab, which was approved for lymphoma in 1997, but quickly adopted for off-shelf use in autoimmunity. And then in 1998, with the introduction of the TNF-alpha inhibitors, infliximab for Crohn's disease, etanercept for rheumatoid arthritis. And since then, there's been dozens of approvals in inflammation and immunology for biologic therapies. In the last decade, we've seen the equivalent concept of targeted therapy that's so well known in cancer come into the space of autoimmunity, with targeting of the kinases JAKs and TYKs, analogous to targeting of imatinib, which revolutionized cancer therapy in 2001. So with that introduction, let's turn to our four experts who are working towards novel small molecule therapies in therapeutic areas where there may or may not already be satisfactory biologic therapies, and discuss the contrasting approaches taken by these companies. I'll start by asking each panelist to introduce themselves and their company, what technologies, biology, therapeutic areas they're working on, and the most straightforward question, why small molecules for autoimmunity and inflammation? If I can start with Natalie.
Thank you very much. Hello, everyone. My name is Natalie Hollis. I'm the CEO of Third Harmonic Bio. We are focused on the development of small molecule inhibitors of the wild-type KIT receptor for the treatment of mast cell-mediated inflammatory diseases. Why small molecules? In the case of the diseases that we're focused on, which are primarily inflammatory and allergic diseases such as chronic spontaneous urticaria, severe asthma, as you say, the current therapeutic options for patients in this space are primarily biologics, and there are a lot of limitations to these approaches, both from actually a safety and efficacy perspective, but ease of administration, and as I'm sure we'll talk about, there's also a need for oral therapies that don't need to be administered in the office. A lot of the diseases we treat, urticaria, for example, is treated primarily by dermatologists. Those offices aren't set up to administer in-office injections. And so as a result, a lot of urticaria patients don't receive treatment for their therapy. So for us, the development of an oral small molecule inhibitor of KIT for these diseases where mast cells are really the primary pathophysiologic mediator of the disease, we think there's an opportunity to really transform the way these patients are managed.
Thank you. Kevin?
Morning, everyone. My name is Kevin Giudice. I'm the CEO of DICE Therapeutics. We are working on small molecules that disrupt cytokine binding to receptors. This has been an area that has historically frustrated efforts to make small molecules, oral drugs, and we've gained insights over the last 5-10 years that are helping us approach these problems successfully for the first time. Our core technology fundamentally is chemistry. but we use a form of DNA-encoded libraries that we think is unique and combine that with insights from structural biology to get a toehold on these historically difficult targets. Our thinking about why small molecules is very similar to Natalie's. There are areas where small molecules just make more sense right now that are serviced by proteins because historically antibodies have had a better time blocking some of these cytokines than small molecules for pretty good reason. But they really should, in a lot of different ways, they really should be small molecules. There should be oral therapies that are more widely available to patients and easier to take. There are other advantages that I think will come with second generation versions of what we're all doing on stage here, and I think they'll play out over the next 10 years, but diseases where penetration may make a big difference, right? So our first indication for our oral IL-17 antagonist would be something like psoriasis, and that's an obvious indication. We know the biology is very well precedented there, but there are other diseases that are very difficult to treat, like hidradenitis suppurativa or HS, where it's pretty clear the antibodies have a tough time at that. And at least one possible argument there is that they don't really get into that severely remodeled skin as well as a small molecule might. So I think there are exciting opportunities in the next gen molecules that we're considering working on that might take us there. So there are a lot of really good reasons to push for small molecule therapies here.
Hi everyone, I'm Rosanna Capello. I'm the co-founder and CEO of Rome Therapeutics. So what Rome's mission is to unlock the dark genome, which is 98% of our genome that does not encode for traditional proteins. And to basically, we believe that it will hold new biology and novel insights that will enable us to develop breakthrough therapies initially for autoimmune diseases and cancer. Our first drug actually is a nucleoside reverse transcriptase inhibitor. It's a potent and selective inhibitor of an endogenous reverse transcriptase encoded by a repeat called line 1. And what happens is that usually when we're healthy, that particular repeat is dormant, so it doesn't go anywhere. But when we are stressed or our cells are basically subjected to irradiation, like sunlight and things like that, repeats get activated and this reverse transcriptase gets expressed. And basically when that happens, it works as a parasite, like a virus, and it can activate our host defense mechanism. And what we have uncovered is that by blocking that reverse transcriptase, we can completely block interferon production in a variety of stimuli. So our first indication is going to be lupus, both cutaneous and systemic lupus. We have seen expression of line 1 in patients with systemic and cutaneous lupus and other diseases, both in the skin and the kidney of these patients. And what is really cool is because as a parasite, it's upstream of all the mechanisms that we right now block with biologics, but that can induce immunosuppression. Because we're upstream of that, what we believe is that we're going to be able to block the initiation mechanism that induces the hyperactivation of the host defense pathways, but we are not going to block the ability of our organism to defend itself from viruses. Because as you know, one of the problems that we have for all immunotherapies right now is that they induce immunosuppression. And then people become more susceptible to viral infection. And as a doctor, it's very hard to regulate that, correct? Because sometimes you're treating the patient for autoimmunity, but then the patient shows up with a life-threatening viral disease. So that's what we are trying to solve for. In terms of why small molecules, I'm a patient. I have autoimmune disease, and I can tell you, injecting a biologic every week or every 4 weeks, it's a pain. And literally a pain. And I think that the other problem is that it's not conducive to combination therapies. So with oral medication, you're going to be able to have an arsenal of drugs that you're going to be able to combine, and you're going to be able to treat autoimmune diseases in a very similar way that today you treat cardiovascular diseases. And I think that compliance is going to go way up once you have these drugs. And the second reason is price. Biologicals are extremely expensive. Basically, there's a whole population of people that don't have access to that. And I think that with small molecules, you're going to be able to bring the price down and enable people access to these medications.
Great. Good morning, everyone. I'm Andre Turan, CEO of Matchpoint Therapeutics. So we're an early discovery stage company, just 18 months old. And our focus, our expertise at Matchpoint is on leveraging covalent chemistry to create, discover, and develop oral medicines that have biologics-like efficacy and selectivity. And the power of covalency really comes from the fact that when you have a molecule that's electrophilic that can engage selectively with a specific nucleophilic amino acid on the target protein, you get some real benefits. You're able to, for the right target protein, get increased efficacy. You can get improved selectivity, and you get a wonderful decoupling of PK/PD that can help lower systemic exposure. So for a number of targets, it's a modality that can open the door to be able to drug targets that have been very, very difficult to access with a small molecule, and in some cases with any modality. So we have a number of programs that we're advancing that are, our lead program is for MK2, and we have also some programs that go after cytokine or cytokine receptors, and very historically challenging protein classes like transcription factors that with a covalent approach, there's some possibilities to finally be able to drug some of these important nodes in the immune pathways. So in terms of adding to what the other panelists have offered in terms of why develop small molecules for immune diseases, I think I agree with all the points made, but I think it also gives this opportunity to, beyond the specific targets that have been clinically validated with biologics, it allows us to explore other targets in the pathways that may open the door for different therapeutic impact. If we go, for example, for our lead program, MK2, upstream of the inflammatory cytokine production of TNF-alpha IL-6, we may have the dual benefit inhibiting the target of impacting multiple of these inflammatory cytokines. So I think that's an additional benefit of a small molecule approach. I think it opens up the range of possibilities.
Thank you. I'd like to spend a little bit more time on the target selection, which we've touched on here a little bit. First and foremost, for drug discovery, target selection really is, a lot of things can go wrong in drug discovery, but if you have the wrong target, nothing's going to work. So I would like to just hear more thoughts about selection of targets. There's a variety of factors that have been discussed here very briefly, but if there's more to say on this area, how you select your targets. I think for some of you, you're in fixed areas of biology, and so the set of targets is small. Others, there's platform capacity here where you have much more freedom over target selection. So,
Do you want us to go in order again, or are we
let's
popcorning now?
We'll do order for a little bit, and then I'll end up selecting individuals.
So the founding thesis around Third Harmonic Bio is that there really hadn't been much advancement in the treatment of allergic diseases for the better part of 2 decades, particularly in the oral space. And the target that we selected, KIT, the biology is very well understood, and as a drug developer, I always like starting with well understood biology because drug development is hard enough without throwing Mother Nature risk into the mix, in my experience. And KIT is very well understood as the master regulator of all elements of mast cell function and survival. And mast cells, which are immune cells that exist in all of our barrier tissues, very well characterized as the primary mediator or primary effector of all of the allergic symptoms that we associate with these types of diseases, swelling, itchiness, and what have you. And so the challenge with KIT, with a small molecule, since it is a receptor tyrosine kinase, is getting the sort of selectivity that you need to target it safely for inflammatory diseases. And that's why it's been a good target for biologics up until this point. And so we had the starting point of a very selective oral small molecule inhibitor for KIT that we pushed forward. We'll talk about the ultimate disposition of that molecule, I'm sure, later in the discussion. But we understood that if we were able to target KIT, we would knock down mast cells, and we would alleviate the symptoms associated with these types of diseases. And one thing that I'll add to the small molecule discussion is in the case of KIT, what we know is you don't need to saturate the receptor. You don't need to completely knock down KIT signaling. You don't need to ablate the mast cell population in order to get good therapeutic benefit here. And one of the advantages of an oral small molecule approach is the ability to dose titrate and really find the sweet spot where you can maximize efficacy but dial down some of the on-target tolerability effects that are known to be associated with a systemic KIT inhibitor. And so not only do we have the advantage of patient convenience, physician preference, all the things that we mentioned previously, but we actually think we've got a better therapeutic approach to this particular target with an oral small molecule versus an antibody.
Yeah, so when we started DICE, we had a sort of technology focus, and we were initially casting about in a wide variety of structural classes of targets, just trying to understand what the technology could do, just what was technically feasible. And we stumbled pretty early on to the idea that we were discovering small molecules that could block certain cytokines, which was quite a surprise, actually. We scavenged around quite a bit in the beginning to find out where the artifact was, and we realized it was legitimate inhibition we were seeing, and then we went about trying to figure out why. And what we discovered was that in the cases where we were having success, we were hitting targets that were cytokines in a dimeric structural class. There are two halves of the molecule. And that started to make some sense to us in terms of looking closely at those structures and realizing that at the interface of these dimers, you will occasionally find pockets that are about the size and shape of a typical small molecule. And that's really the starting point you need to have a good shot at doing what we're all talking about doing here. You need some kind of a leg in. So when we found that, then we basically went about and said, okay, that would be really neat if we could do that consistently, because that's evaded chemists for a long time. So let's go and look at that universe of dimeric and trimeric proteins and then try and figure out where are their therapeutic gaps. So, there are plenty of those proteins in all therapeutic areas, but the one that really struck us was immunology because, as my colleagues have said so eloquently, that's an area where small molecules really are preferred for a variety of good reasons, and simultaneously they just had been absent, largely absent for 20, 25 years because the targets are really hard. And so you had this sort of paradoxical thing where you have all these great biologics in immunology, and they tell you exactly what the right target is, but it's not the right modality. So we narrowed our focus to immunology and then took off from there. And it's been a really interesting ride. We're going to have an R&D day coming up actually next week, and we'll unveil some work on new targets. But it's looks like it's going to be quite a fertile area. And what I like about the approach we've taken so far is that, and Natalie alluded to this, by choosing a target that's well validated, we've taken a lot of the risk out, which for a small company is very, very important. Trying to keep an organization like the 4 companies up here funded and moving forward, there's a lot of risk. And we chose to pull the risk up to the things that we thought we could adjudicate best as a company of 15, 20 people in the beginning. So we're chemists. We think we can do chemistry pretty well, but we don't bring anything special to the table in terms of biology. So we're not going to get set about to validate targets as our first mission, right? Choosing the wrong target going into phase 3 can be lethal for a company, almost any size, but certainly a small company. So we're avoiding that. But we're setting ourselves up in the 2nd and 3rd generation efforts at DICE to go into more ambitious spaces, so intracellular targets behind the blood-brain barrier, and then eventually we'll get to brand new biology. But for now, we're very happy where we are.
Rosanna?
So we're a little bit different because we're working on a novel biology, a novel target. So how did we come to that, correct? Why a reverse transcriptase? And the reason for that is because there's vast epidemiological data in patients with HIV showing that as they age, they have a decreased incidence in a variety of autoimmune diseases, sometimes up to 80%. And that got us very curious. Why is it? These patients are not infected with viruses. Why is it that by taking antiretroviral therapy, they don't have autoimmune disease. And we were interested in the dark genome or the repeatome, this area of the genome that's comprised of repetitive sequences that are derived from viruses that have integrated in our genomes through evolution. So we said, maybe there is something there that we should look at. And we did, and there are actually two elements. Line 1 is one of them that encodes for reverse transcriptase, and the other one is human endogenous retroviruses that also encode for reverse transcriptase. So the question for us was, okay, so what happens if we block those reverse transcriptases? Can we actually phenocopy what has been seen in the clinic? And when we're asking this question, a paper came out in Nature from Yannick Krau's group showing that in patients with Aicardi-Goutieres syndrome, so these are kids that are born with autoimmune disease, it's a rare genetic disease, and basically they resemble patients with very severe lupus and viral-like encephalitis. And what he did is he treated these kids with pediatric antiretroviral therapy and was able to show that he could decrease the interferon scores in these patients. And I remember calling him up and I said, oh my God, this is amazing. And we are starting to develop a nucleoside reverse transcriptase inhibitor for endogenous reverse transcriptases. This was 3 years ago, and he said to me, call me back when you have a brain penetrant compound. Because in these kids, what you really need was a brain penetrant compound, which is not available because nucleosides usually don't cross the blood-brain barrier. So with all the data, we basically realized that the reverse transcriptase inhibitors that exist today are not potent and selective enough. So we started doing our own chemistry. My whole group, before coming to Rome, I was the founding CSO of Nimbus Therapeutics. So one thing that we know how to do very well is chemistry. So we said, okay, let's take what we know and do it well. And now we have a potent and selective inhibitor of line 1 reverse transcriptase. So we also discovered that HERV-K reverse transcriptase, it's not the culprit here, is line 1 reverse transcriptase. And we can show now that we have very potent and selective compounds that can completely block interferon pathogenic interferon. It doesn't touch interferon when it basically is activating by viruses, but completely blocks this pathogenic interferon that's involved in autoimmune diseases. So that's how we got with the target selection. In addition to that, which is a platform, there are two other areas of the dark genome that we're interested in. One, this we call the viral mimicry pathway. So you have a parasite, an endogenous parasite that basically activates the host defense mechanism. But the other two things that they do, they also can make antigens. So this is another area of research. And the third piece is that they can also induce genomic instability. So we are looking at all three areas of biology. Andrea.
Well, thank you. That's a actually inspiring story of the kind of reverse translation of not the bench to bedside, but bedside to bench.
Absolutely.
And a good reminder of how clinical observations can provide insights that can be quite driving and for biology and drug development.
So that's how we de-risked, if you will, this biology because of the clinical data that was available. That's why we de-risked the biology, because can you imagine founding a young company and going through completely novel biology? It was going to be too much of a high risk.
That is a risk. Natalie highlighted the preference for going into therapeutically validated areas for drug development because of that
Especially
risk.
these days when the financial environment is tenuous. It's really difficult to argue for new on top of new.
Yes.
There you go.
Yes.
So I think this was the other thing that we liked is that reverse transcriptase inhibitors have been used in people for over 40 years now. We know how to manage them. We know how to administer them. It's an oral medication. We understand that like the first generation of reverse transcriptase inhibitors have issues with mitotoxicity, mitochondrial toxicity. So this was one of the first screens that we put in place. So we were informed of 40 years of human, experimentation, if you will.
So
in
Andre,
terms
you have a very exciting technology that could be widely applied for many targets in therapeutic areas. So very excited to hear your thoughts on selection of targets in therapeutic areas.
Yeah, thanks. Similar to Natalie and Kevin, we look to lower the biology risk we take on with the covalent approach a certain degree of chemistry risk. So whether we have some biologic validation of the given target, as I mentioned, of the pathway, or sometimes just very strong, in some cases, bidirectional genetic evidence of the involvement of the target that's not been drugged to date. That's how we sort of think about the degree of biology risk we take on, but we look to lower that. The second factor for us as we think about targets is thinking about the rationale for covalency. So it's not every target that is suitable or where covalency is needed. It's an irreversible engagement of the target. And if, for example, the target has a very short half-life, it may not be a benefit to have this irreversible type of engagement. So we think target by target about what is the anchor rationale for covalency in our prioritization of targets. And for example, MK2 as our lead program, that's a kinase, and we're targeting the active site. And as with many kinases, you have a strong affinity in that binding pocket for the natural ligand, ATP. So this is an area, this is a place where if you have an ability to have full target occupancy with an irreversible binder, you may have the ability to displace this otherwise natural ligand that has strong affinity. And we also have the benefit with MK2 that it's a very long half-life protein. So this element of having the benefit of coming in and out, but having the small molecule attached to the protein for the full life of that protein against a protein half-life that's prolonged, that's a strong benefit. So that's how we look to marry the technology with target selection at match point.
Well, you have an interesting set of the 3 targets you mentioned, ranging from novel targets to the IL-4 receptor, where you're going to directly compete with dupilumab head-to-head, with a very established, effective mechanism. So I'm interested in that kind of mix, whether that's perhaps in part business-oriented, that's something a company needs to do, or how you arrived at both novel targets as well as a tried-and-true target.
Yeah, so we look at a target like IL-4 receptor alpha as being one, again, that's creating a lot of value for patients across now, I think, 5 approved indications. But there still are a number of patients that are non-responsive or lose response. And the point that Rosanna mentioned, I think, is an important one. I think in the future, combination therapy for many of these chronic conditions will be feasible when we have the ability to have combination oral therapies. So there's a number of factors that make this a very, very compelling target to try to have an effective oral therapy. And on the backdrop of our criteria, once again, so you have an issue, a challenge that Kevin touched on with cytokine and cytokine receptors. In this case, it's the affinity between the ligand and its receptor that makes it challenging for a small molecule to find an anchor. So with our covalent screening approach, we're able to find and have discovered binding site that interfere with this complex. So it gives us an ability to intervene. So I would say it's a superb target in terms of biological validation, and one we think is suitable for the tool that we bring to the table.
No one's going to complain that oral Dupy is a bad idea, I think.
Correct. Yeah.
Generally speaking.
And just to riff on those themes a little bit, Stephen, I completely agree with what Rosanna said. Combo therapy is definitely going to be the wave of the future in immunology, and I think what you're looking at here are 4 approaches to really rifle shot into targets that we think are going to be great, right? So, IL-4 definitely is going to work for AD. You don't have to worry about that. 17 is going to work for psoriasis. But if you think about
Kit's going to work for urticaria.
Kit's going to work for urticaria.
Thank you. Yeah.
Thank you. My bad. But if you think about something that's more complex and arguably more prevalent, like rheumatoid arthritis, right? You look at the history of biologics in that space, lots of things work a little bit. The gold standard is Humira, anti-TNF therapy. But even there, only about 50% of the patients are going to respond, and only 50% of them are going to have a durable response. So there's clearly an opportunity there to mix and match the way that has been so successful in areas like cancer and infectious disease. But as Rosanna pointed out, you're not going to be mixing and matching injectables. It's just not going to happen for any of a variety of reasons. But if you get each of us is successful, which I think we hope we all will be, then you've got 4 targeted therapies that you can start playing around with. And there are 6, 8, 10 other people who could be sitting on this stage with us, all working on pretty targeted therapies. 10 years from now, those things will be combined. Shrewd observations by clinicians will make it happen. And that's when things are going to really start opening up. So this is step 1. Step 2 is going to be innovative in ways that are more biological and more clinically oriented. We're innovating now, I think, heavily in the technology approach right now, but it's coming. There's going to be a lot to think about here.
And when you're combining oral small molecules, you can dose titrate. There's more,
Absolutely.
you can be more nimble in the way that you're combining those therapeutic approaches, whereas with a biologic, it's a shot and you sort of
And you can
cross your
remove it
fingers.
also.
And
you
fast
can
on,
take it
fast
away.
off.
Adverse event, you can just remove it.
That's right.
Whereas with the biological, it's much more difficult to do that.
Yeah.
I just want to make a plug for the JAK inhibitors and TYK inhibitors. So these are the new oral molecules, correct? I mean, the JAK inhibitors have been used for a while now. They are efficacious, but they have a lot of adverse effects, whereas the TYK2 inhibitors seem to be as efficacious as the JAK inhibitors, but with less side effects. So it's going to be also very interesting to see what the TYK2 inhibitors will do to the arsenal of drugs that we have today to treat a variety of autoimmune diseases, including, for example, psoriatic arthritis. You know, because that is an area that there is very little out there for. You know,
Certainly in the oral space, yeah.
Exactly.
That's right,
exactly.
yeah. And JAKs are used there sometimes, but they're...
But the problem with the JAKs
boxes,
is the, exactly.
Yeah, I mean, the boxes... Yeah, no, I think there's a lot of innovation in that space. There are a number of TYK2 companies, all of whom seem to have pretty good drugs. So it'll be interesting to see how that plays out. But it does feel like we're in this renaissance period of small molecules and immunology after a long period of not much happening.
And it's better for patients, more options,
For
more
sure.
accessibility to therapy. There's always more to be done.
That's right.
Will be interesting to see how the tick story plays out. It is early days. Some of the side effects of the JAKs weren't emergent until post-marketing, such as the thromboembolism and so forth. Right. So, but they do seem like a very promising class of medicines. So, related to, certainly therapeutic area is quite linked to the target. Targets naturally define therapeutic areas, but I think most of you, if not all of you, perhaps have some other choices outside of I&I, even that where you could be going for mast cell neoplasms, for example. Getting into oncology, which is often a flip side of autoimmunity. So I just wonder, briefly, if any of you are also considering oncology areas or other areas outside of I&I for your technologies. Please.
We are both oncology and neurodegeneration. So there is actually quite a lot of literature now pointing out that our first target, line 1 reverse transcriptase, can play an important role in neurodegeneration. And then there is a lot of work being done for the viral mimicry pathways and also the genomic instability in oncology. Okay. But it's more complicated because it's not only about the tumor itself, but also about the tumor microenvironment. So trying to understand that it's more complex than in autoimmune neurodegeneration.
And Natalie, over mast cells?
We're really focused in inflammation and immunology. We're focused on wild type KIT for now, and most of the mast cell driven oncology indications are driven by mutant KIT. But even within immunology, there are 10 different diseases that could be amenable to a KIT inhibitor. So from my perspective, when I think about company building and expertise building and being able to do that in a focused and efficient way, again, I'll say in a time when the financial markets are tight and you really need to be disciplined about the way that you're investing, There's a lot to be done in the immunology space that will leverage the existing expertise that I have, not only in chemistry and preclinical development, but importantly as I move forward into clinical development. But that's my company. There are different ways to skin the cat, but for Third Harmonic, we're focused in immunology.
Yeah, I think there are a couple of factors that play in here. One is that, so for example, one of the early hits we had at DICE was a small molecule that disrupts PD-L1. So some obvious applications there, right? But as we sat and thought about it, we had two reactions. One was exactly what Natalie just said. We're not going to be experts in everything, and being expert even in I&I is going to be a challenge. And then the second thing I think is more subtle but interesting, which is for diseases like oncology and arguably infectious disease, what you're looking for is a breaker switch. You want to take this, move it from here to here and shut something down. With most immunological diseases, you want to think of it more as a rheostat, right? You want to dial it in, you want to titrate the dose, you not necessarily want to shut the whole immune system down for any period of time. So there are different challenges, and that sort of breaker switch can often be accomplished by an antibody. Now, where things get really interesting is intracellularly, right? If there are signaling molecules intracellularly that are not kinases, not easy to shut down, those could be interesting challenges. But for us, they'll be a later generation. We're really focused on I&I, and there's plenty to do there.
Yeah. In covalent C, a lot of the successes in the past 10 years have been in oncology. Recently, Lumakras, KRAS, and we have the BTK, GFR examples that where you have some superb selectivity for a mutant over wild type. So there's a lot of nice work done there. Again, our focus for now is completely on immunology. We think there's no reason why you can't have highly selective covalent medicines that are used chronically. And in many ways, we can take a page from the experience in oncology by being able to leverage a unique nucleophile on a protein of interest, sometimes to achieve that selectivity over a closely related homologue or an anti-target. So there's an approach here from the experience in oncology, many ways that when we look for target selection to match the modality, we can bring to the space of immunology.
I think that in our case, we're letting the science drive the indications so that we're letting the targets speak to us in terms of what we should be going after. And we're taking a slightly different approach where we see that we can have a franchise around one specific target, whereas the molecules themselves are going to be very different, because for neuro, you need brain-penetrant molecules. For oncology, you need to shut down the target completely, whereas for immunology, you only need to shut down maybe 60 to 80%. So the molecules are going to be very, very different properties, So that is why, I don't want to shut it down, but I do agree with Natalie that's very hard to do that in a small company. So the way to actually do that is to partner. Mm-hmm. So we are looking into partners for both neurodegeneration and oncology, and we internally will focus in autoimmune.
Well, in the last minute or so, I do want to touch base on another distinction between small molecules and biologics, which is the predicted toxicity. Biologics have relatively predictable off-target toxicity as well as metabolic toxicity, which tends to be less predictable for small molecules. So I'm just, wondering about how you all think about this. I know Natalie has had an experience in the phase 1B.
We can't end on drug-induced liver injury. We can't spend the last minute of our panel talking about the downsides of oral small molecules. But we live with chemical risk. Drug-induced liver injury it hangs over all of us, and it's a risk that is often not predicted by extensive preclinical work, and you don't see it until you get into the clinic. It happened to us last year. The way you mitigate that risk is with chemical diversity. Backups and backups to your backups. Luckily, that was a part of the business that we invested in from the very early days, and so we developed a hypothesis for what we saw. We tested it. We had molecules that addressed that hypothesis and tested those, and you pick yourself back up and you keep going because you believe that what you're doing has a place in the market. patients out there can benefit from what we're doing. Value can be created for patients, for shareholders. We can build a great company around this idea. You just pull yourself up by your bootstraps and keep going.
Great. Well, it's really spectacular science that you're all engaged in. Very, very exciting therapeutic molecules that are being created. And we're very thankful for the session today and for your frank discussion.